Hello everybody, welcome back to another exciting edition of Ed Puzzle Lecture Notes. Today we're going to go over enzymes and the chemical reactions that they can catalyze or speed up. So what is a chemical reaction? It's a process that changes one set of chemicals into another set of chemicals simply by changing the bonds between the atoms. So right here you can see a chemical reaction, CO2 plus water gives you this that you drink in your sodas every day. That's carbonic acid. So basically when you put CO2 with water together, they react to form this very weak carbonic acid. Now what you need to know is the things that go into a chemical reaction, so these are the ones that are going into the reaction, those are called the reactants. Reactants. So right there. That's what goes into this chemical reaction. We got carbon dioxide and water. Those are our reactants. And the things that get produced or made, those are the products. So in this case, we're producing carbonic acid. So the reactants go into the chemical reaction and the products come out. And our daily objective. So this is your objective of the day. When you walk out of class, you should be able to do that, or do this. You should be able to describe how enzymes speed up chemical reactions in the body. So enzymes speed up the chemical reactions in your body. You also should know what a substrate is, what an active site on the enzyme is, and last but not least, you should be able to tell me at least two things that can change the enzyme's shape. And if an enzyme's shape changes, the enzyme becomes worthless. So, how do enzymes speed up chemical reactions? What is a substrate? What is the active site? And what are two things that can change the shape of enzymes? That is our objective for today. Alright, so now here's a couple broad classes of chemical reactions. The first one is called endergonic. Endergonic reaction. That's what RXN stands for. It stands for reaction. Endergonic reactions are reactions which absorb or store energy. So these are not reactions that blow up. These are actually reactions which store energy. For example, you can see right down here. This is photosynthesis. So you have energy from sunlight striking the leaf, and the plant actually stores the energy from sunlight and stores it in this molecule, sugar. And precisely it stores it in the sugar glucose. So that would be an endergonic reaction because it is storing the energy from sunlight and putting it into a nice sugar molecule, which then can be released later, and that energy can be released from the sugar. Now, the opposite of photosynthesis is an exergonic reaction, exergonic, and this is where the energy is released. So this is the very opposite of photosynthesis. This is respiration, and this is where glucose, that same sugar that we made in photosynthesis, well, now we're going to break that sugar molecule down, and this is what's releasing energy, and it's in the form of ATP. ATP equals energy. So that would be exergonic because it's releasing the energy from glucose. So that energy can be used by the body. So endergonic stores energy. Exergonic releases the energy. All right, so I'm going to focus mainly on what's really important. So chemical reactions, we have these things called catalysts. Catalyst is a substance that speeds up the rate of a chemical reaction, and it does it by lowering the activation energy that is required. So what is activation energy? That is simply the energy that's needed in order to get the chemical reaction started. So sometimes you need a little energy to kind of kickstart it. It's like in cartoons, you know, the bombs in cartoons like Bugs Bunny, they always look like a bowling ball with a fuse sticking out. All right, well that fuse, once it's on fire and it burns down, that's the activation energy required to set off that little cartoon bomb. Well, the same thing. Catalysts, they speed up the rate of chemical reaction by lowering the activation energy. So this is a little example. There's a barbecue pit. Now you could hold a match there on the little briquettes, the charcoal, and wait, or you can speed up. You can speed it up by adding some lighter fluid. That'll cause the fire to start much faster. So that's what a catalyst is. It's a substance which speeds up chemical reactions. Catalysts speed up chemical reactions. And here we are to the enzymes. Super important. Enzymes. So what do you need to know? First, their names. They're called enzymes. Number 2, they're made of proteins. So these are special proteins which act as biological catalysts. Remember, a catalyst is anything that speeds up chemical reactions within the cells. So in enzymes, they're made of proteins, and they're catalysts which speed up chemical reactions within your body. And once again, these enzymes, they work. They make the reaction go faster because they lower the activation energy that is needed for the reaction to occur. But the main thing you need to know, they're made of proteins and they speed up chemical reactions in the body. Now let's see if you can remember this. What are the monomers which make up proteins? So enzymes are made of proteins. But what are the monomers that make up proteins? Let's see if you remember. What are the little guys that you put together to make proteins? All right, hopefully you told me that the monomer of proteins are amino acids. All right. Okay, so now it's time for a little brain break, and I'm going to ask you a riddle. What word, when you add 2 letters to it, actually becomes shorter? So what word, when you add 2 letters to it, actually becomes shorter? Think about it. And the answer, of course, is short. Because if you add 2 letters to it, E and an R, it becomes shorter. All right, now back to enzymes. So here we go. This green with a weird shape there, that guy's an enzyme. You can see he's got a very strange shape. And it's this guy's shape that means everything. Now these are the reactants. Remember, the reactants are what goes into a chemical reaction. So here we have 2 reactants. They're going to bond with this enzyme. Now, in a reaction that's catalyzed by an enzyme, our reactants have a new term. They are called substrates. Substrates. So these 2 guys right here, the red guy and the blue guy, those are 2 substrates. They're the reactants. Now, whatever gets made from this enzyme, that's going to be the product. Now, where the substrates fit in perfectly on the enzyme, like this little spot right here, has a perfect shape for this guy to come fitting in. And then this little spot also has a perfect shape for this guy to fit in there. That spot that has the perfect shape for the substrate, that's called the active site. Active site. So the active site is the spot on an enzyme where the substrate is going to bond to it. And then eventually these 2 guys are going to bond together. All right, so once again, here's an enzyme at work. This gray thing, that's the enzyme. This triangle and whatever that thing is, those are the substrates. Those are the reactants which are going into this chemical reaction. Now you can see here, this enzyme has a perfect shape. Right there. That one's kind of like a triangle. Well, that substrate, that triangle, that orange triangle is going to fit in there. And same thing with this guy. It's going to fit in that exact spot right there. Remember, that's called the active site. The active site's the spot on an enzyme that the substrate fits into. And now this enzyme kind of plays the role as a matchmaker. So like your friend that tries to set you up with a date. Well, this enzyme is going to set up this guy right here and this girl right there. Now, eventually those two could have met each other just by accident, but the matchmaker makes it happen much quicker. So the substrates bond on to the active site, and then boom, they leave together as a couple. So that chemical reaction combined those two substrates into this green product over here. Well, enzymes can also be that guy or that girl that doesn't set you up, but breaks you up. So, these two people are boyfriend and girlfriend or whatever, and he likes to break them up. Well, that's this enzyme right here. Same thing, protein. It's got an active site. You can see the active site right there where these substrates are going to bond on. And this time, the enzyme doesn't bond them together, but it actually splits them up into the two products. So here we have the substrate coming into the reaction, and we have our products that are leaving. So the main thing to get from this is, what are substrates? What are products? And what is the active site? If you can answer those three things, good job. All right, we are almost done. Enzymes. So I want you to think of enzymes like this key right here, this car key. So what you need to know, I could take my keys and try to start all the cars at Tulare Western, but it's not going to work because my key only works with one type of car, my truck. Same thing with enzymes. Enzymes can only catalyze one type of reaction, so they don't do everything. So for example, lactase. That is an enzyme that only breaks down the sugar lactose. So that lactase breaks down lactose. It does not break down sucrose. It does not break down maltose. So just like a key, these enzymes are very specific. They only work on one type of reaction. But also like a key, every time I get home from school at night, I don't throw away my key and buy a new one. Because guess what? Keys can be used over and over and over and over again. Well, same thing with enzymes. Enzymes can catalyze reactions over and over and over again unless its shape is altered. So if I took my car key and I stuck it in a bunch of hydrochloric acid and it totally melted my car key, guess what? It wouldn't work anymore. And the same thing is true of enzymes. If you change their shape, they no longer work. So the thing to remember, it is the shape of the enzyme. Because remember, they have to fit specific substrates. Whatever their shape of the substrate is, that enzyme has to have the same shape so their substrates will fit into it. And now the last thing, what are two things that can change the shape of enzymes? What can change the shape of enzymes? The first one, extreme temperature. So if it gets really, really hot, extreme temperature, like it gets over 200 degrees, that can destroy the enzyme, that can change its shape. The other thing that can change its shape is extreme pH. Remember, the pH goes from 0 to 14. So most enzymes, they like it somewhere in the middle. They like it somewhere around 7. Remember, that's neutral. Most enzymes don't like it to be really acidic, like a strong acid with like a pH of 1. Or they also don't like it to be very basic, so like say a pH of 13. Both of those extreme pHs would change the shape of the enzyme and make the enzyme worthless. Okay, last slide. Why is an enzyme and substrate compared to a lock and key? Well, it's simple. Because remember, the substrate has a specific shape which fits into the active site of that specific enzyme. So that key, that substrate, will only work with this specific active site. And this lock will only work with that specific key. Same thing with enzymes. They can only catalyze or speed up one specific type of chemical reaction. They can't do all the different types. Question number 2, what happens to the enzyme after the reaction? Well, that'd be like asking, what are you going to do with this lock after you open it? You don't have to throw it away. Enzymes can be used over and over. You can keep reusing them as long as their shape doesn't change. And that's why is the structure of enzymes important? Because it is the shape. Just like the shape of a key opens that lock. If the shape changes, that enzyme will no longer work. And what are 3 things that can alter the shape of an enzyme so it doesn't work anymore? I'm just going to go 2 because that's all we need. Remember, extreme temperature, like really, really, really hot. That can change the shape of an enzyme. And the other thing that can change it is extreme pH. So that just means very, very acidic. So like a strong acid. That can change the shape of an enzyme. And also a very, very strong base can change the shape of an enzyme. All right, thank you for watching. I hope you could understand most of it, and I'll talk to you next time.